Welcome back to another episode of Concentrating on Chromatography. Jacob, thanks so much for being our guest today. I really appreciate it.
Thank you for having me.
Would you mind introducing yourself and telling us a little bit about your background?
Definitely. So I'm currently a third-year PhD student at the University of Washington in Seattle. I'm studying chemistry, specifically analytical chemistry and mass spectrometry. So we focus a lot on glycoproteomics. I'm in the lab of Professor Nick Riley, and we focus a lot on method development for mass spectrometry-based glycoproteomics. Before I came to Seattle, I was an undergraduate at The Ohio State University in Columbus. And there I worked in the lab of Professor Susan Olisic. And I did really broad work in her lab, focusing on enhanced fluidity liquid chromatography. They're using liquefied CO2 in the mobile phase to improve efficiency of separations. But then I also dipped my toes in electrospun nanofibers for something called SALDI, the surface affected laser book mining, as opposed to MALDI. I really enjoyed my undergraduate research, and that led me to.
Very cool. And for the first time ever, I have a co-host today. Candi, would you mind introducing yourself?
Yeah, my name's Candi Gokey. I'm a, fifth year PhD candidate at UCSD and SDSU. I study chemistry, but I use a lot of method development for untargeted metabolomic studies. I've studied a lot of the gut microbiome and how diet impacts overall human health, and my current project is not related to any of that. And it's actually elucidating how corals and algae talk to each other. And I'm super stoked to talk with Jacob because one large part of my process of my current study is understanding how glycoproteins and fragments of glycoproteins are moving through liquid chromatography mass spectrometry to see if we can see any of those as artifacts and that are being mischaracterized as artifacts in the mass spectrometry data sets that we're looking at. So I have lots of questions.
Excited to talk.
Yeah,
Yeah. It's a fairly new topic, too. People weren't looking at glycoproteins or glycoscience until, about 10 years ago or something like that? It's not something that's been readily studied because it's not overly dictated by DNA. It's largely secondary metabolism that's dictating how those proteins, glycoproteins, are being structured. And correct me if I say anything wrong, because my specialty is the method development part, not necessarily the chemistry part. So that's
and Jacob, just to start us off, could you talk about how you got into mass spectrometry and specifically glycoproteomics?
For sure. It really started when I was an undergrad. I was a chemistry pre-med student and obviously didn't end up in med school. And in my second year, and I did my degree in 3 years, so this was pretty late in my studies. I was working in the hospital system at Ohio State, and I was working in the radiology department, dealing with patients very directly, and I enjoyed it for the most part, but about 1% of the interactions were not the best. And even in those cases, you obviously still have to deal with the patient. There's no real way outside of that interaction. I didn't love that aspect of it. And at the same time, I was taking quantitative analysis, and that was taught by a professor named Amanda Humman. And she's actually a mass spectrometrist. She focuses a lot on mass spectrometry imaging and proteomics, specifically for colorectal cancer applications. And I thought that was just really cool, the fact that you could apply exactly what we were learning in the classroom to something as real world and as important as colorectal cancer, something that has touched my family's life, but so many other people's lives. And was I enjoyed that class so much that I loved doing the homework. There was no other class before that where I loved doing the homework. And that was the first class that I went to office hours. I had an awesome conversation with Professor Humman, and that really sparked my interest in analytical chemistry. So I knew from that point forward I wanted to go to grad school. I also didn't know at the time that you get paid to go to grad school and you do not pay tuition to go to grad school. So that also influenced my decision a bit. Yeah, it's awesome, right? And I knew I needed to get into a research lab. I was asking professors left and right. The one professor who welcomed me into her lab was Professor Elisik. And from there, dipped my toes in a little bit of mass spec, and I absolutely fell in love.
That's great to hear.
Oh, and then I didn't touch the glyco side of things. Yeah, so there, I graduated in 2023. That was the year that Professor Carolyn Bertozzi won the Nobel Prize in chemistry. And I watched her Nobel Prize lecture. And again, I was fascinated by real world application. And that was, again, cancer. The fact that cancer cells become what we call hypersialylated. So they have a lot of sialic acids. The way that she was describing it was very elegant and extremely fascinating. And I began digging into the literature on glycoproteomics because I knew that I had at the time been accepted into grad school at the University of Washington. And I noticed that there was an incoming professor, Nick Riley, and was digging into his research. There's actually a Glycoproteomics Nature Methods Reviews primer on glycoproteomics, and I was just digging into that and again fell in love with the subject. And I knew that I wanted to join his lab.
For listeners who don't work with glycans every day, why is glycoproteomics such an important yet difficult area?
So, the first thing that I always bring up is the fact that every living cell that has ever been studied has a coating of glycans, and we call this the glycocalyx. And glycans play fundamental roles in so many facets of biology. So, everything from the immune system, it plays huge roles in the immune system. And of course, the immune system is involved in cancer, among other diseases, of course, but we focus a lot in the Riley group on cancer. But glycans also play roles in protein folding and protein stability. And the list really is endless on how glycans play roles in biology. Now, it's definitely not easy to study glycans and especially glycoproteins. So, what people, when they're studying glycoproteins for a long time, what people have done to make it easier is they enzymatically remove the glycan from the protein. And this is not the best approach because now you're losing the information as far as what amino acid on that protein is decorated with a glycan. In the Riley Research Group, we do intact glycoproteomics. We keep the glycan attached to the protein, but this can be challenging for a number of reasons. And it's literally from every step of the analysis that we face challenges in glycoproteomics. And it is really an evolving field. Everything from sample prep to data acquisition, and we'll talk a bit about that today, but even after you acquire your data, there are bioinformatic challenges. And all of these challenges were really focused on addressing in the Riley research.
Before we get into a lot of the more technical terminology and stuff, if somebody was really interested in looking at your research and going into the topics that we're going to discuss, what are the three fundamental topics that they would have to understand in order to go into more depth of what we're going to talk about today?
Yeah, that's a really great question. I would say you need to know at the core what a glycan is. So there are two main classes of glycosylation. There are N-linked and O-linked glycans. And what that stands for, it's nitrogen and oxygen linked. So I think knowing like what the two classes of glycans are is very important when you're getting into the field because for a long time, and I still think currently, it's treated as almost two separate fields or two subfields, if you will. And so that's number one. Number two, if we're thinking specifically about mass spectrometry based glycoproteomics, I think the way in which people use mass spec, the types of technologies that we use in mass spec or glyco, would be very important to know. Something that we think a lot about is the different ways that we can fragment our glycoproteins in the mass spectrometer. That's very important. And then I think some of the things that you see in your data, once you finally acquire it with those technologies, something that we think a lot about is how glycans fragment in the mass spectrometer. and the resulting fragments. And some that we'll actually talk about today, they're called oxonium ions. They're these glycan-specific fragments. And knowing what those are is also very important. So those are the...
And something you both know about is method development. Jacob, what problems kept frustrating you enough that you decided that new methods were needed?
I would say that new methods are still needed in glycoproteomics, and that's the challenging thing. I think that glycoproteomics really does lag a bit behind traditional proteomics when we're thinking about things like how many IDs we're getting, or even if we're thinking about other PTMs like phosphorylation, issues that we face in glycoproteomics, like enrichment, like how do we selectively enrich for glycoproteins, glycopeptides after we entry with an enzyme versus sampling the rest of the proteome. That's a challenge that we face. And like I said, we face challenges at every stage of the analysis. And I think that can maybe make people less excited about the field. But for us, it really does make us more excited because these are more opportunities to push the field forward. So there's just so many considerations, and we really focus on addressing these problems so that we become a little bit more, it's more of a level playing field with, say, traditional proteomics.
Are you guys focusing more on instrumentation or more on the post data analysis, like cleanup?
That's a great question. So we actually have worked at, I would say, really every stage of analysis. So we have some folks working on the enrichment side of things, two grad students who I can think of, Kayla Markuson and Vanessa Encinas. They're both working on some enrichment strategies. We have a postdoc, Tim Veth. He's working on some more post-acquisition data analysis, also a grad student. Katie, she's working on some more tools to analyze our data, for example, GlyCounter, which is out there. So we really do focus on every step from instrumentation, sample prep, and then data analysis.
And you had briefly touched on the differences between N-linked and O-linked glycosylation. Was there anything else you wanted to say about the different fragmentation strategies that they each require?
Yes, that's a great question. First, I'll point out N-linked glycosylation occurs at an asparagine residue. And following this asparagine residue, there's some amino acid that's not proline. And then following that amino acid is either a serine or threonine. So that's highly specific and a highly conserved consensus motif at which N-glycans occur. Now, for O-glycans, it's a completely different story. They can occur at any serine, threonine, or even sometimes tyrosine. And there's no known consensus motif. We often will employ different fragmentation modalities for N-glycopeptides and O-glycopeptides. So for N-glycopeptides, because we have this such conserved motif, and then on top of that, there's often only one of these motifs in a triptych peptide. And then on top of that, we often will see peptide fragments that keep that very first monosaccharide of the N-glycan intact when we use collision-based methods. So there we're colliding our ions with a gas. So for all of those reasons, we can get away with using collision-based fragmentation for N-glycans. Now, for O-glycopeptides, because they can occur at any serine or threonine, we need to have peptide fragments that keep the glycan intact. And if we use collision-based methods on O-glycopeptides, we're going to lose the glycan from the peptide. So there would be no spectral evidence to assign the glycan to a given amino acid. And you can have multiple of these O-glycans on a single peptide. And we often do see that, and that further complicates things. So to get around this, we use an electron-based strategy. So there we use specifically electron transfer dissociation because we use thermo instruments. And there we're taking an electron reagent anion and we're reacting it with our analyte cation. And when we do that, we are specifically fragmenting along the peptide, and we're not fragmenting off the glycan. So it's pretty soft on the glycan. And there we have peptide fragment ions that keep the glycan, and we have spectral evidence to assign what amino acids are modified by glycan. Now, to further complicate things, we actually use ETD followed by supplemental collisional activation. So we actually use both when we study O-glyc, and that's something that we call ETHCD. So it's just never-ending acronyms, really. And the reason we do this is because there can be some non-covalent interactions that kind of keep our fragment ions sticking together. And so we use just a little bump, if you will, with collisional activation to get those apart from each other. And then on top of that, we do a little bit of glycan fragmentation with collisional activation as well. That's where we get our oxonium ions. So if we had only done ETD alone, we wouldn't see those oxonium ions, and they can provide us a lot of information in our spectra as well.
And historically, my understanding is that researchers often had to choose between optimizing for N-glycopeptides or O-glycopeptides. Why is that exactly?
That's a great question. And it's almost like people still do this. And I think it's the different challenges that each face. So everything from enrichment, how well are we enriching for N-glycopeptides and O-glycopeptides at the same time? I think that's a challenge that we continue to face. But then from the mass spec side of things, people, again, with these different fragmentation modalities for each, these different optimal fragmentation modalities, you have to choose what method you're going to use for each. So previously, people tried to do simultaneous N and O glycoproteomics, and kind of the pioneering method was something called product-dependent triggering, where we look at a collision-based MS2 spectrum, look for these oxonium ions to tell us, is this a glycopeptide or is this not a glycopeptide? And then from there, if you see those ions, then you would trigger ETHCD on the same precursor. So we're still at an MS2. And the issue there is you're asking the question, is this a glycopeptide? You're not asking the question, is this an N or an O glycopeptide? And there, you're wasting time taking ETHCD scans that these scans take more time than HCD, right? Because there's a chemical reaction happening between that reagent anion and the analyte cation. And I just have to stop for a second and just say how cool that is. The fact that we're doing chemistry in our mass spectrometers like that, that's really cool. But it takes more time is the drawback with it. And so we want to optimize so that we get as many scans as possible we use a 90-minute LC run. So we're running on a 90-minute gradient, which is quite long, especially for people who think a lot about traditional proteomics. So since it is so long, we want to make sure we're getting as many scans as possible. That's kind of a drawback with those product-dependent methods, is you're now triggering ETHCD on every likely glycopeptide rather than optimizing between the two.
Do you see a difference in the number of fragments or your ability to be able to identify N-link and O-link when you're using positive or negative mode? Or do you guys strictly just run in one or the other?
That's a great question. So we mostly will do positive mode. I think when you start getting into negative mode, that's especially when you're thinking about doing glycomics. This is, again, where we enzymatically cleave the glycan from the peptide and then just analyze the glycan. And there's some benefit to running in negative mode there. And what you'll often see is we actually want to use glycomics to inform our glycoproteomics. So we're actually getting closer to this idea of glycomics assisted glycoproteomics, where we actually do both. But yeah, I would say we mostly run in positive mode. Now, in terms of fragmentation of N versus O-linked glycopeptides, the main difference, I would say, or there are actually a couple of differences. One is you'll see these peptide fragment ions that retain that first monosaccharide of a glycan. That's called GlycNAc. So we actually represent these as fun shapes with colors because it just makes our life a lot easier. So GlycNAc is a blue square whenever you're looking at cartoons of these glycans. And so the peptide will retain that very first blue square. Glycans don't do that. So we often will just cleave off the entire glycan in collision-based methods. So that's one main difference that we see in terms of fragmentation. Another is kind of intensities of these oxonium ions. So we'll see differences in relative intensities between certain oxonium ions when we fragment versus glycans.
And your group developed autonomous dissociation type selection. What is that, and what were you trying to solve with that?
Yeah, when we talked a moment ago about optimizing for N versus O glycopeptides, again, where people use the product dependent methods, what we were trying to really address was the drawback of doing that. And instead of asking the question, is this a glycopeptide spectrum? We instead wanted to ask, is this likely an N or an O linked glycopeptide spectrum? And the way we do this in autonomous dissociation type selection, or ADS, is using something called real-time library searching, or RTLS. Again, we're going to see so many acronyms. And the way this works is it's actually quite simple in terms of how we're using RTLS. So calling it a library search is perhaps a bit of a stretch because we have two reference spectra, and that's it. That's what makes up our library. And what we're doing is as we're collecting data, we're telling the instrument to look at these spectra. We actually use product dependent triggering before RTLS so that we're only running RTLS on likely glycopeptide spectra. And RTLS takes these two reference spectra, one's for N-linked and the other's for O-linked, and we're looking at two specific fragment ions. These are 138 and 144. So I mentioned before GlycNAc, that's blue square. We see that a lot in N-glycans. O-glycans, we often will see just GalNAc. So that's a yellow square. It's an isomer of the same class of monosaccharides called hexNAc. So these are all, hexNAcs are all squares, and then the different shadings tell you what isomer it is. And 138 to 144 is a GlycNAc to GalNAc ratio. So it can clue you in to how much GlycNAc or GalNAc you have. So for N-glycopeptides, we see far more GlycNAc in the glycan. So the 138 to 144 ratio will be very high. And then for O-glycans, it's roughly 1-to-1. And that's just based on how the different glycans fragment. And we'll take those two reference spectra, compare it to the spectra we're actually collecting, and we're only focusing on those two ions, which is what makes it a relatively simple strategy. And if it matches that more 1-to-1 ratio, we would call that a likely O-glycopeptide. And then from there, trigger ETHCD only on O-glycopeptides. We're only taking time for ETHCD when we really need it and not wasting it on glycopeptides.
And one thing that's been so revealing about these podcast conversations is that it seems like every researcher has a novel use of either AI or machine learning. So I'd love to hear about your application of machine learning and some of the advantages that it provided you over, I believe you were originally using real-time library searching. So anything you'd like to share about that would be great, Jacob.
For sure. That's an excellent question. So something I didn't mention about ADS is there is a drawback, and that is that we are optimizing for most O-glycopeptides by using this relatively simple strategy with real-time library searching. The reality is some O-glycans can contain GlycNAc. And when they do, that will make our 138 to 144 ratio appear more like an N-glycan. So in those cases, we are not triggering ETHCD, even though they are O-glycopeptide spectra. And so we're losing out on that information. And these glycans do play important biological roles. One thing that you actually see this in is the intestine. So at homeostasis, you see the intestine being lined with these core 2 and core 3 glycans that contain glycnac. And that's actually an interest of mine because in an inflamed state, in the intestine, you actually see less of these. So it's almost like the glycnac's getting stripped away. So understanding these differences and being able to see these differences in an inflamed gut mucosa versus the homeostasis really is important. So we wanted to use machine learning because here we would be able to take into account all of the oxonium ions. So rather than just focusing on 2, we're looking at around 50 oxonium ions at a given time. And that's just the power of something like machine learning. If I wanted to do that, I would have to empirically determine the intensity ratios for all of these different types of glycopeptide classes. And that would just take a very long time. And it's actually something I know because I've tried looking at 3 other oxonium ions. So these are 407, 569, and 528 to try to look at those intensity ratios. And it was very difficult. And part of a challenge that we face, again, in glycoproteomics is we don't have a lot of standards. So again, one thing that sets us apart from proteomics. So having something like machine learning, we can take published data sets that are relatively well annotated and train machine learning models on those and the oxonium ion intensities in those data sets. And from there, train the model to take into account all of those oxonium ions rather than just looking at most 5 oxonium ions. And that really does provide you with a bit more confidence in these classifications.
In untargeted metabolomics, we face all the same challenges that you just mentioned too. So fairly new science too. So I work with Dr. Peter Dorstein's lab, who works closely with Ming, and they're in UCSD and UC Irvine. They do the Global Natural Products Social Molecular Networking, GNPS. They have a whole suite of metabolomics data sets that they are building for like workflows that they're building, addressing these same problems. And one of the tools that I'm using specifically for my research is this program that they call MassQL. And essentially, I can create this library of fragments or like specific mass to charge ratios that I'm looking for and search all of the metabolomics libraries that are uploaded in GMPS. So I was super interested to know how far you guys are with that and if you guys are using any pre-existing models that are out there to adapt to your guys' references, or like if that was something that you think would be helpful to with your guys' project because it's already an existing structure that's successful, and like merging those, it's all in GitHub. So I was just kind of curious, are you guys handwriting all of your stuff, or are you guys pulling from other sources, or how are you guys doing all that?
That's a really interesting idea. And we are training these models from scratch. So it would be really useful to have something that's already existing to pull from like that. But yeah, currently what we're doing is we're taking published data sets, and it can be difficult to find a data set with all of the data that you need. That's just the state of the field currently. One that we've been using is actually quite a large data set. So they're claiming like 200,000 in glycopeptide IDs and 50,000 O-glycopeptide IDs or so. And that's a very expansive data set to pull from. But as far as training the model, that's all done from scratch. So it would be really useful to have something to pull from for sure.
Are you guys using any in silico fragments or anything when you're doing your testing? Are you like throwing weird stuff in there that's just like, we know this doesn't exist, or maybe it does exist and it finds it?
No, that's a really interesting idea. And not currently, but I think that's something to look into for sure. Yeah, currently, I guess I should mention how we're pulling these oxonium ion intensities. So we're using in-house software called Glycounter, which I mentioned very briefly before. And what it does is it extracts oxonium ion intensities and puts it into a very nice spreadsheet. And from there, really what we're doing is we're taking that spreadsheet as input data for a machine learning algorithm. And specifically, I've been using something called XGBoost, which just so happens to be developed at the University of Washington. I didn't know that until after I had started using it. But yeah, that's currently how we're training the model, is using GlyCounter, and extracts all of those intensities for us. And we're actually moving toward using it in real time as well.
How long does it take to get through one round?
Yeah, so as far as if you're looking at a large data set and running it offline, the GlyCounter search is probably the longest part. It can take less than 10 minutes or so, depending on how large the data set is. And then XGBoost is extremely fast. Like, I'm talking seconds to train an entire very large data set. And then to actually run it again, it's on the order of seconds, if not faster. Whenever we're running it in real time, the really nice thing about XGBoost is it's on the order of milliseconds. So that's what's enabling us to be able to do it in real time. And I should point out, what we're doing there is we're extracting the intensities of oxonium ions by running not only XGBoost in real time, but we're running GlyCounter in real time as well. So it's first GlyCounter is taking those oxonium ion intensities, extracting them in real time, and then feeding it to XGBoost. And then XGBoost makes its classification. And the model is pre-trained, so we don't have to worry about training the model in real time. And then that whole process is on the order of milliseconds, which makes it really nice for running in real time.
That's pretty awesome. That's very fast.
Was there a specific moment you could point to, Jacob, with your application of machine learning where you said to yourself, oh, this actually works better than we even hoped for?
For sure. So I should say we did work in collaboration on this project. We worked with Bill Noble's lab here at the University of Washington in the Department of Genome Sciences. And I worked very closely with a grad student named Justin Sanders. And they knew a lot more about XGBoost than I ever did starting this project. And they helped out a lot with training the model initially. And the point at which I thought this would work well is we worked together on a manuscript, which is currently pre-printed. And there they were looking at a couple of different machine and deep learning strategies to classify glycopeptide spectra. And then we compared it to ADS using RTLS. And we benchmarked these different methods against each other and showed that for a large published glycopeptide data set, all of the machine and deep learning strategies outperformed by far ADS. And I thought that was very exciting. And then getting deeper into using XGBoost on other data sets, something that I realized is the fact that XGBoost is classifying core 2 O-glycopeptide spectra, not as N glycopeptides, but now as O-glycopeptides. So now we're able to recover that information using XGBoost, which I think is very exciting.
And do you foresee a future where some of these intelligent acquisition strategies become more standard components on mass spectrometers in general?
I really do hope so. The way I view intelligent data acquisition is squeezing the most out of our instrumentation. And I really do think it's synergistic between scientists, whether you're thinking about researchers in academia or in industry. It's really synergistic between us and the instrument vendors. So I really do hope that instrument vendors more broadly support real-time mass spectrometry and continue to expand support for real-time mass spec, because it really does benefit all of us to use real-time mass spec. It gets the most out of these instruments, and I think it's a very exciting space. And I think what's even more exciting is my hope is that people see what is happening in the real-time mass spec space and then thinks of ideas themselves and gets to work on implementing them, because that's what's going to push the field forward. The more people that we have making these methods and coming up with these very cool ideas is the most exciting part for me.
Yeah, I definitely agree. The real-time mass spec stuff is a big push because it's like you can learn so much more about not only your data set, but also what's actually happening in the instrument. Because I don't know if you guys do any of the native MS stuff, but that's a lot of an area that would be really great for doing the real-time analysis, because you're running this whole protein through the mass spec. And if you're doing science, sometimes in the mass spec, right at they use a lot of ESI. So right within the ESI droplet, doing chemistry right at the point. So being able to identify what's happening in there would really be beneficial for the real-time identification and library searching. And I think like a lot of the vendors are actually like trying to implement that, but we're currently limited with manpower and computing power, and like the cloud has been really helpful, but like it's expensive, and I think we'll get there. I think it's like the onset of how the Orbitrap was conceptualized before it was actually able to be fruitioned. Like I think we're not going to have as long as a gap between the two things, but it's really exciting time to be in the field. It's just hoping funding can keep going.
Oh yeah, for sure. And yeah, that's a really interesting point about native mass spec. I haven't really thought about real-time mass spec in that space, but that is a very good point. I've done some, whenever I was like empirically thinking about oxonium ion intensities, we had a somewhat of a standard that I was working with and directly infusing it into the mass spectrometer. This wasn't native MS per se, but there I can definitely see the value, because you are changing these instrument parameters in real time, and it can be difficult to know unless you set all of this out from the very start. But sometimes maybe you see something in your analysis as you're infusing something. You're like, that would be interesting to do an experiment on, but maybe to have real-time mass spec to guide you there could also be helpful.
And Jacob, my understanding from your work was that you applied a pretty novel approach for intact glycopeptide analysis. What gave you confidence that would work ahead of time? And could you explain what some of that work was?
For sure. Here we're shifting gears and thinking about something called polylacnac in glycans. So these are very elongated in glycans. They have this repeating chain of galactose and glycnac. Galactose, we resemble it as a yellow circle. So you have this repeating chain of a yellow circle and a blue square. And it makes your N glycan just extremely large. And they play huge roles in biology. Again, thinking about the immune system and cancer, this can be one way by which cancer cells can evade immunity and eventually metastasize. And so they're very important to analyze, but they're very challenging. And one reason that makes them so challenging from a mass spec perspective is they can be poorly ionized. The ionization efficiency can just drop because they are so large. And then on top of that, they have such a high, what we call microheterogeneity. So this is the way in which the monosaccharides are assembled into the full glycan. And you can maybe think about, if you have a very long chain of these repeating monosaccharides, maybe you have 4 branches on your N glycan. What's to say some of those monosaccharides are on one branch, or maybe they're across the 4 branches? And the way that we think about glycan structure, it's really all inferred within glycoproteomics, although we have a bit of work being done on structural glycoproteomics with some real-time methods. But currently, we infer things from glycan compositions. So that makes it challenging when you have this very long repeating unit. So what we have started doing is enzymatically removing that long chain. So not removing the full glycan, we still keep the full glycan intact, but we removed the portion of repeating units of monosaccharides. And that simplifies the analysis, because now we're able to ionize it. And then on top of that, there's far less microheterogeneity there. We do this using an enzyme called endo-beta galactosidase. It's actually an enzyme that's endogenous in many cells. And what it does is cleave the galactose that begins on that branch. And what you're left with is an N-glycan that's partially galactosylated. So each of these branches, if you think about a tetraantenary N-glycan where we have 4 branches, each of those 4 branches would have a galactose near the end, except for the repeating unit, which starts with the galactose. If we cleave off just before that galactose on the repeating unit, the lacnac chain, then we're left with one branch that has no galactose. And from there, we can infer from glycan composition because we're missing a hexose, we're missing a galactose. And so that makes it a bit easier for the analysis. Now, as far as what gave me confidence that it would work, we were working in collaboration with a couple of different labs for a project that I can't get into detail about because it's currently under review, but they had been doing it for glycomics. So we knew it worked on the glycan level. It's just we didn't know that or no one had done intact glycoproteomics work with this enzyme. But the fact that it worked with the enzyme did definitely instill confidence that it would work on intact peptide level. It's just more what kinds of things would we have to think about from a method development perspective in terms of when do we use this enzyme and things like that in the pipeline, that those were more open-ended questions.
I always like to say that whenever you're ready to come on and talk about that next project, I'd love to have you back on the podcast for another discussion.
For sure.
And Jacob, as we get short on time, I wanted to ask, what else about your work do you want to make people aware of? Or was there anything else that's absolutely critical that you want to share that we may have glossed over?
So on the same line of things with polylacnacylation, something I can share is some of my own work on cutaneous versus uveal melanoma. So this is a really interesting application that we're using our developed endo-beta galactosidase workflow on. Now, I actually never heard of uveal melanoma before I started this project. So uveal melanoma, it's of course a cancer of melanocytes in the eye. And it's really a grim diagnosis because it metastasizes distantly at a much higher rate than cutaneous melanoma. The metastasis rates are about 5% for cutaneous melanoma versus 50% for uveal melanoma, thinking about distant metastases. And the majority of those metastases in uveal melanoma occur in the liver, so far removed from the eyeball. And then thinking about survival rates, the 5-year survival rate for cutaneous melanoma, it's about 35% compared to less than 20% for uveal melanoma. So I think that's really a stark difference there. And thinking about how there are these differences in metastasis, that got us to thinking, how could polylacnacylation play a role here? Because we knew from before starting this that polylacnacylation is involved in tumor cell invasion and migration. Another thing I'll point out, uveal melanoma, there's only one approved therapy specifically for uveal melanoma. It was actually in the last few years. It's called tebentafusp, and it's marketed as Chemtrack. It's only indicated for about half of the patients with uveal melanoma because it requires a specific HLA marker. And it's a weekly therapy. And for the first 3 weeks, patients need to be hospitalized because they can experience some severe side effects from this therapy. So the field definitely has a far way to go. And we're hoping that our work on polylacnacylation can help these patients a bit.
No, it's super critical work. Absolutely. Jacob, thanks so much for joining the podcast today. And Candi, thanks for co-hosting and asking wonderful questions. I think the audience will get a lot out of this episode, and I appreciate you both for being here.
It was great meeting you, Jacob.
Hey, you too. Thanks for having me.